All right, good evening, everybody, and welcome to Manhattan Community Board 6's COVID-19 Vaccine Town Hall. 1 year ago, none of us could imagine the destruction that this pandemic would bring to our communities in 2020. Most notably, our most vulnerable populations, the elderly, people of color, and those with pre-existing conditions, succumbed to the virus disproportionate from the rest of the population. On top of all of this, economic devastation forced many to continue to commute to work and put themselves in harm's way, and exposed other socioeconomic divides. All in all, as of tonight, the most updated statistics show that New York City has seen more than 500,000 cases of COVID-19, with almost 26,000 deaths. Amidst this darkness, many in our health and science fields have been working nonstop to fight, manage, and eventually eradicate COVID-19 as a deadly virus. We are proud of the healthcare workers in our district, particularly at Bellevue Hospital, the Manhattan VA Medical Center, and Tisch Hospital at NYU Langone, as well as all essential workers who have worked tirelessly and at their own risk to ensure the health and safety of our communities. When I was elected board chair in November 2020, one of my first priorities was to establish a standalone Health and Human Services Committee to focus solely on the pandemic and to address our community's physical, mental, and emotional health and well-being. To serve as chair of this committee, I have appointed Dr. L.V. Barroso, who has served on CB6 for 5 years and is currently a professional development coordinator for the New York City Department of Health and Mental Hygiene's Bureau of Tuberculosis Control. Her credentials include a medical degree, Master of Science in Occupational Medicine, Diploma in Epidemiology and Tuberculosis Control, Master's in Public Health with a specialization in Community Public Health, Master of Science in Nursing, and because that wasn't enough, a PhD as well. Dr. Barroso has 33 years of experience in public health, 28 years of experience in conducting trainings, planning, organizing, and coordinating educational activities, and 12 years of experience in teaching in New York City nursing schools. Furthermore, she is a certified infection control trainer by the New York State Department of Health. To ensure that our community board is a think tank and the source for accurate and reliable information in our district, I ask that the first meeting of our revamped Health and Human Services Committee be a town hall on the COVID-19 vaccine. There is a lot of misinformation and rumors circulating around this topic, particularly given the politics of the moment, which is to the detriment of us all. I am pleased that we are joined today by leading experts on the topic of vaccination and infectious diseases from world-renowned institutions, such as the Johns Hopkins University Bloomberg School of Public Health, the NYU Langone Vaccine Center, as well as the New York City Department of Health and Mental Hygiene. We are also pleased to be joined in attendance by Michael Stinson from the New York City Comptroller's Office, Pat Hill from Councilmember Keith Power's office, and Bob Gormley, who's the District Manager of Community Board 2. We thank you for your attendance tonight. As we proceed with tonight's town hall, I urge all to listen intently, to ask your questions, and to share the information and knowledge gained from tonight's program with your communities. Furthermore, today serves as the first of many conversations, programs, and initiatives to address the health concerns of our district. I encourage all of you to continue to work with our Health and Human Services Committee, which will be held regularly every second Thursday of the month. Members of the public, you can raise any questions or comments that you have through the Q&A feature of Zoom. Health and Human Services Committee members, you can raise any questions or comments you have by raising your hand. Once all of tonight's panelists are done speaking, and once we field questions from Health and Human Services Committee members, we will answer questions submitted through the Q&A feature. So once again, I thank you all for your attendance and participation, and I look forward to a lively and informational discussion tonight.
Thank you, Kyle. As Kyle mentioned, my name is Dr. L.V. Barroso. I'd like to thank everyone watching at home for tuning in tonight, and I would especially like to thank for our wonderful speakers for tonight. Our first speaker tonight is Dr. William Moss. Dr. Moss is the Executive Director of the International Vaccine Access Center and a professor at the Departments of Epidemiology, International Health and Molecular Microbiology and Immunology at the Johns Hopkins Bloomberg School of Public Health. He will now give us updates about the COVID-19 vaccines.
Thank you, Dr. Barroso, and it's my pleasure to be here. I'm just going to share my screen. All right. So what I'd like to do to start off this town hall, and again, I thank the organizers for inviting me and giving me this opportunity to provide a very brief overview of COVID-19 vaccines. I realize that some of you have a medical background and have more understanding of the COVID-19 vaccines, but I'm going to address this to the general public, but hopefully it'll be of interest to everyone. So the COVID-19 vaccine development, as all of you know, has been a truly remarkable achievement. It normally takes 5, 10, sometimes even longer years to develop a vaccine and get a vaccine out into the public. This has been done with COVID-19 vaccines in just under a year, a truly remarkable achievement. And we need to pause and be so grateful that we're even having a discussion about having highly effective and safe vaccines 1 year into the pandemic. Certainly we could have used these vaccines earlier. We are in the midst of a tragic pandemic, but it is a remarkable achievement in the history of vaccines in order to have such a protective vaccine at this time. What I'm going to do is go through some of the basic types of COVID-19 vaccines and then really focus in on the messenger RNA vaccines, the mRNA vaccines. You may know them as the Pfizer or the Moderna vaccines, and talk briefly about some of the characteristics, both the efficacy and safety of those vaccines, talk a little bit about what we don't know yet and what we still hope to learn, and then go through some frequently asked questions. And obviously we'll have time for questions from you after all of the speakers have given their presentations. I thought I would start off by just reviewing very briefly some of the typical types of vaccines so that you can understand how the messenger RNA vaccines differ from these typical ones and how they're similar. So there's a long history of developing vaccines. Vaccines owe their origin to Edward Jenner just at the end of the 18th century, who used cowpox to vaccinate against smallpox. But the real advances in vaccinology came actually with Louis Pasteur in the mid-19th century. And some of the techniques that were developed early on in vaccine development are still used today. Although certainly with much more modern techniques. So one way of developing a vaccine is by inactivating a virus. And some examples of these inactivated viral vaccines include the vaccines we use for poliovirus, for hepatitis A virus, and for rabies. Basically, what's done here, and I'll show you an example for SARS coronavirus 2, the virus that causes COVID-19, is to use a chemical or radiation to basically take the virus and make it such that it cannot replicate. Some people call these killed viruses or replication deficient or incompetent viruses. We'll call them inactivated viral vaccines. So that's an old method and kind of standard method of producing vaccines. Another relatively typical way of making vaccines, but quite different, is to attenuate the virus. And so examples of attenuated viruses include measles virus or rubella or mumps, often combined here in the United States as measles, mumps, and rubella vaccine. These are attenuated viral vaccines. What that means is the virus is weakened, but not inactivated or killed. So these viral vaccines can still replicate within our bodies. And in fact, that's how they induce a strong immune response. But instead of taking either the whole virus and killing it, or the whole virus and making it attenuated, we can use just parts of the virus. We can use certain proteins from the virus. And I'll give you an example for SARS coronavirus 2, but some typical examples include the vaccines for human papillomavirus, HPV vaccine, or some of these toxoid vaccines like for tetanus or diphtheria. These are using a modified protein, so it's just a part of the virus or the pathogen. In the case of tetanus and diphtheria, it's a toxin that's produced by a bacteria. Now, more recently, there have been advances in what I'm going to call gene-based or genetic vaccines that instead of injecting the protein or the whole virus, we actually inject the genetic instructions to make part of the protein. So instead of using or injecting the protein, our bodies, our own cells, particularly muscle cells, make the protein that then our immune system responds to. There's only been, prior to these COVID-19 vaccines, only one previously licensed vaccine, and that was for Ebola virus, and that was what we call an adenovirus vectored vaccine. And I'm going to talk more about that in relation to COVID-19 vaccines. But I just wanted to start off by giving you kind of an overview of different types of vaccines that are already in use before the pandemic. Now, I'm going to rely on a lot of images from the New York Times. They've put together a very nice compilation on their website called the Coronavirus Vaccine Tracker. And I've kind of taken some of their images and kind of put them together into slides to show you how various types of COVID-19 vaccines work. But first, I wanted to start with this summary. This is from yesterday, and highlight the fact that there are a lot of COVID-19 vaccines in development in the pipeline. We've had 2 in the United States that have been issued an emergency use authorization by the United States Food and Drug Administration. Those are the vaccines that are being used now. But there are 65 different COVID-19 vaccines in early development, and an additional 20 vaccines in late stage development. So there are a lot of COVID-19 vaccines in development. We have never seen in the history of the vaccine development this many vaccines produced in such a short time for a single pathogen. So let's talk a little bit about how these different COVID-19 vaccines work. We're going to start with the basic SARS coronavirus 2. I know many of you have heard about this, but these are relatively simple. They're called RNA viruses. It's called coronavirus because of these spike proteins that stick out of it that make it look like a crown on an electron micrograph. So on the inside of this virus, you can see that coiled RNA. That's the genetic instructions to produce the proteins for the virus. And you can see highlighted there the spike protein gene. That's going to be important in our story as we talk about how these different vaccines work. And then obviously on the surface of this virus, you can see the spike protein. That's what we're interested in developing our immune response to, and I'll describe that in more detail. But all the COVID-19 vaccines in development are really targeting the spike protein on the surface. The reason why is because it's that spike protein that binds to receptors on other proteins on our cells and allows the virus to get into our cells and then replicate. Viruses cannot replicate on their own. They can only replicate within cells. And it's the SARS coronavirus 2 uses the spike protein to get into our cells. So I'm going to start a little backwards. I'm going to end with the messenger RNA vaccines because I think it's helpful to kind of build up to that and talk about some of these other more typical vaccine platforms, which are not yet approved for use in the United States, but are in development elsewhere. So first, let's start with this inactivated viral vaccine. There are a number of pharmaceutical companies and vaccine manufacturers around the world that have built these inactivated COVID-19 vaccines. Basically, you can see the virus there. They use a chemical, beta-propiolactone, but you can use different chemicals. And as I said earlier, this is to inactivate the virus so that the virus kind of stays whole, but cannot replicate. So the way these inactivated COVID-19 vaccines work, and I'm going to give you a little bit of immunology. This may be new to some of you, but basically that inactivated coronavirus is taken up by what we call an antigen presenting cell. That's a fancy name for a cell that kind of eats up the virus and presents viral proteins on its surface to other cells of the immune system. So that's what's being shown here. It's taking up and it's showing on its surface, kind of displaying that spike protein to what we call a helper T cell. A helper T cell then these will divide. You'll get in the immune response, there'll be a lot of these helper T cells formed, and they help the B cells produce antibodies to the virus. And that's the main mechanism through which our COVID-19 vaccines work. So what's being shown here on the right side is this helper B cell that's been activated because it's seen the spike protein from the antigen presenting cell. It's showing it to this B cell that's going to make the antibodies. And then as you'll see, these antibodies then bind to the spike protein on the surface of the virus and block the virus. These are what we call neutralizing antibodies. Once these antibodies bind to the spike protein on the surface of the virus, that virus can no longer enter our cells and it basically dies and is cleared. There's also, so this is fundamental, this process, this immunologic response is fundamental to all COVID-19 vaccines, producing these antibodies that neutralize the virus. There's also a cellular immune response that's shown here on the right side that activates what we call killer T cells. So we have helper T cells and killer T cells, and these killer T cells will kill infected cells. So we actually have two mechanisms for getting the virus out of our body, one by neutralizing antibodies that prevent the virus from entering cells, and also by activated killer T cells that kill infected cells. So how do protein COVID-19 vaccines work? So I just explained how inactivated COVID-19 vaccines work. There are in development protein COVID-19 vaccines, one by a company actually here in Maryland named Novavax. They produce the spike protein, actually very interesting, in moth cells. That's what's shown on the left-hand side. It's just a system for generating proteins. And then they take those proteins shown here and they stick them onto very small particles called nanoparticles. So you get kind of a cluster of these spike proteins. And then they add what we call an adjuvant. It's basically a chemical that helps stimulate our immune response. And so that's how these protein vaccines work. And these are now in phase 3 trials here in the United States. So we may see these protein vaccines kind of coming along in the pipeline in the coming months. And they basically work the same way. These proteins or clusters of proteins on the nanoparticles taken up by the antigen presenting cell, shown to these helper T cells, stimulating them, activating them, and then they in turn will activate the B cells to produce the antibodies. that then bind to, in the same way I showed before, the virus and prevent the virus from entering our cells. Now, the adenovirus vaccines you may have heard about. These are a little more complex vaccine platforms. There's been work on these vaccines for many, many years, but as I said, the only licensed adenovirus vaccine was for Ebola virus. That was in 2019. But you've heard probably in the news about the Oxford University AstraZeneca vaccine. That's an adenovirus vectored vaccine. It uses a chimpanzee adenovirus. The Johnson & Johnson or Janssen vaccine has been in the news recently because they released some preliminary study results that uses a human adenovirus as the vector. And then in Russia, the Gamaleya Research Institute, many people have been vaccinated with another type of adenovirus vectored vaccine. Now, these adenovirus vectored vaccines are different in that they are the first that we're talking about of a gene-based vaccine. Here, the gene is in the DNA form, not the RNA form. And basically the adenovirus vector, they're made replication deficient. So these adenovirus vectors cannot replicate themselves, but basically they're a delivery, they're a packaging of the genetic instructions that goes into the nucleus of our cells. That's converted into messenger RNA shown over here. And basically our own cells, our muscle cells, then use that genetic instruction from messenger RNA to produce the spike protein that's released from the cells and stimulates the immune system in the same way that I've described. So this is just another way of presenting the spike protein to our immune system, but using another virus, adenovirus, to provide the genetic instructions to our cells to produce the immune response. And then lastly, I'll just talk about the messenger RNA vaccines. So these are the BioNTech Pfizer vaccine, the NIH Moderna vaccine, the two vaccines that are now in use in New York City and in the United States following the emergency use authorization in December from the Food and Drug Administration. Here, instead of having an adenovirus deliver the genetic instruction, we have the messenger RNA itself. And it's wrapped in this kind of fatty lipid envelope to protect it and to allow it to get into our cells. But basically what happens is that genetic instruction or recipe gets into our cells produce from that genetic instruction the spike protein that's shown on the surface of the cell and released from the cells when they're killed, and our immune system then responds to the spike protein of SARS coronavirus 2. This is a technique, a technology that people have been working on for more than a decade, but these two vaccines, the Pfizer and the Moderna vaccine, are the first time we've had an approved and widely used messenger RNA vaccine. So basically, it's a similar process. This is just showing those antigen presenting cells taking up the spike protein, activating the helper T cells, and then the B cells make the antibodies. And when those antibodies increase in number, this process takes about, can take a couple weeks for our antibody response to really build up, then we're protected. And if these antibody producing cells remain for a long time, what we call memory B cells or plasma cells, then we have long-term protection because they're building these antibodies continuously over time. And when our body sees the SARS coronavirus, we can have a very rapid immune response. So that's how these vaccines work. Now, I want to talk a little bit about the efficacy and safety of these vaccines. Just very briefly, I know there'll be questions around this. This is a complex slide, but the bottom line here is that the Pfizer and the Moderna messenger RNA vaccines have very similar and very high protective efficacy. as we learned in what we call these large phase 3 clinical trials. And I know you're going to hear more about clinical trials later in this session. But basically, 30 to 45,000 people were randomly allocated by chance to either get the mRNA vaccine or a placebo vaccine, and then they were followed over time, and the investigator measured whether or not they got COVID-19. And that's what's shown in these two curves, here for the Pfizer vaccine, here for the Moderna vaccine. The top line is the placebo group, and this is a cumulative number of cases in the placebo group, and the red line is the vaccinated group. And you can see over time, these two lines diverge, showing the protective efficacy of the vaccine. We see the same thing with the Moderna vaccine. I think many people, including myself, were very surprised, happily surprised, at the very high level of protection showed in these large phase 3 trials of these two vaccines. As I said, 95% after the second dose. The endpoint, clinical endpoint in the Pfizer trial measured that protective efficacy 7 days after the second dose, and the Moderna vaccine 14 days after the second dose. I'll just say this little inset here shows these two curves separating right after the first dose. So there is some protective efficacy after the first dose of these vaccines, but we don't really know. These studies were not designed to really look at protective efficacy after a single dose. They were designed to look at protective efficacy after 2 doses. In terms of side effects, we learned a lot from these large phase 3 trials, and we're learning more as more and more Americans are being vaccinated. I just made this table here from the phase 3 trials showing the different side effects that occur roughly within 12 to 36 hours of vaccination. Many people have heard about these side effects. They are worse after the second dose than after the first dose, but these, what we call reactogenicity or inflammation, are really a consequence of our immune system responding to the vaccine. You can see they're quite common: soreness at the site of injection, fatigue, headache, muscle aches, chills, joint pain, fever. There are some slight differences between the two vaccines, but roughly comparable in terms of these these side effects that are occurring. Some people need to take a day off from work, particularly after the second dose. Some people treat these with anti-inflammatory agents like Tylenol or Advil. You all may also be aware that as more and more people were vaccinated, first in the UK and then here in the United States, there have been some people, rarely, but some people have had a severe allergic reaction, what we call an anaphylactic reaction. This is, we're still learning about these side effects, these more severe side effects, these allergic reactions. It probably is to that fatty layer, that fatty envelope that protects the messenger RNA. There's something called one of the components there, a polyethylene glycol. There have been reported previously people with allergic reactions to polyethylene glycol, and that's the leading candidate. There's actually not much to the ingredients of these vaccines, both the Pfizer and the Moderna. It's the messenger RNA, and we don't develop immune responses to that, at least allergic reactions. That's a normal part of our body. There's these fatty layers, and then there are some salts and buffers. So what do we still need to learn about these vaccines? So the emergency use authorization really came after a preliminary analysis of the phase 3 trials. These phase 3 trials are still ongoing, and there's still much to learn. But I also want to emphasize that we already know a lot about their protection. We know a lot about their safety, and no corners were cut, no shortcuts taken in terms of conducting these trials. And again, we'll talk more about the trials later. But we still don't know the duration of protection. We're very early in this vaccine rollout. We're still following individuals in these phase 3 trials. So we need to learn about the duration of protection. I expect it's going to be at least a couple years. I don't think we'll need annual vaccines, but we will need to see. How long? It's possible that there will be a need for booster doses, maybe at 5 or 10 years. I'm just guessing there, but there may be a need. We need to learn more about the safety of these vaccines in pregnant women. There's no reason to think that they're going to be unsafe in pregnant women, but obviously that's of concern and we need more information. The phase 3 trials deliberately excluded pregnant women, although there were some women who became pregnant during the course of the trial. We need to learn more about vaccination of children and the safety and efficacy and dosing in children. The Pfizer vaccine has an emergency use authorization for individuals 16 years and above, the Moderna for 18 years and above, but the clinical trial data thus far does not have information on children. So that's an important thing that we're going to learn in the coming months. We need to know more about the protective efficacy in people who are immunocompromised, whether that's for some underlying condition, HIV infection, or cancer chemotherapy. Those people were included in the clinical trials with those underlying conditions, but we need to learn more. The numbers were relatively small. And then there is always with a vaccine, a potential for rare adverse events. And we only learn these as we vaccinate millions of people rather than tens of thousands of people. So there's still more to be learned. We don't have all the answers yet. I'll just conclude with some frequently asked questions. Is one vaccine better than the other? These Pfizer Moderna vaccines are very similar in their efficacy and safety profile. They do have differences in their cold storage. The Pfizer vaccine requiring that ultra cold temperature that the Moderna vaccine does not. There are slight differences in the age distribution or target age group, as I said. And the Pfizer vaccine does need to be reconstituted with a diluent prior to injection. Will the current vaccines cover the new variants? This is a very important question. There's still ongoing work on this. I don't expect that the virus has evolved away from immune protection from these vaccines, but we're going to need to see. And as I said, I think we're going to get some answers, at least preliminary answers, for the UK and the South African variants in the next week or so. Can one still get the virus after being vaccinated? Yes. These vaccines are not 100% protective, and as I said, 95%. And some people may be able to get asymptomatic infection and potentially even transmit. We just do not know yet. The phase 3 trials looked at clinical disease, not asymptomatic infection. When does the vaccine become protective after administered? Well, the best data we have from the phase 3 trial is that people are protected 1 to 2 weeks after the second dose. But I did show you that there is some protection even after the first dose, although we do not yet know how long that protection lasts. So the best data we have is 7 to 14 days after the second dose. What happens if you don't get the second dose within the time frame? Well, this too is an important question. The data that we have from the phase 3 trials is where the second dose was given on time. 3 weeks after the first dose for the Pfizer vaccine, 4 weeks after the Moderna vaccine. I don't think from an immunologic point of view, it would make a huge difference if the vaccine were delayed a week, 2 weeks, 3 weeks, but we just don't have the information to say what the impact of that is. Does a person who got sick of COVID and recovered still need the vaccine? The current recommendations are yes. The immune response, the protective immune response to COVID-19 is variable. We do not yet know how long that immunity lasts. And the general recommendation now is even if you've had COVID-19, you should get the vaccine. Some experts say you should wait 90 days after the infection to perhaps allow for the immune system to respond to the vaccine, but that's not clear and not all experts agree with that. When is the ideal time to get the vaccine? after a person has recovered from COVID-19 symptoms, and I just addressed that. It's unclear. Some experts feel that it can be, that one doesn't have to wait. I know Dr. Tony Fauci suggested 90 days, but we really don't have the data on that. So I'm going to stop there. I think we're a little over time, and we can move on to the next speaker.
Thank you, Dr. Moss, for a very comprehensive presentation. Your presentation increased our understanding of COVID-19 vaccines. Our next speaker is Dr. Rupali Limay. Dr. Limay is the Director of Behavioral and Implementation Science at the International Vaccine Access Center and is a full-time faculty member in the Departments of International Health, Epidemiology, and Health, Behavior and Society at the Johns Hopkins Bloomberg School of Public Health. She will now speak about issues relating to vaccine hesitancy.
Thank you so much, Dr. Barroso, and it's great to be here. I'm very excited to be able to present to you all. Hopefully everyone can see my screen okay. Okay, great. So as Dr. Barroso mentioned, I'm going to focus on a phenomenon called vaccine hesitancy, and I'm going to really focus on communication and trust. So I think Dr. Moss had a very similar chart just in his presentation with regards to updates as to where we are with potential candidates for a vaccine. And as Dr. Moss mentioned, there are hundreds of candidates that are still going through various phases with regards to the trials. The good news is that here in the United States, we do have 2 approved candidates. And again, as Dr. Moss also mentioned, that both candidates have very similar with regards to effectiveness, with regards to 95% effectiveness in both from the clinical trial data. But the question is, and I say this a lot, I've been working on vaccine acceptance for the last several years. And now that a vaccine is available, it's very important for us to be able to communicate to the public that vaccine recommendations really reflect the state of scientific knowledge. So this has been challenging for us in a number of ways within the context of the COVID-19 pandemic, as well as COVID-19 rollout, which has started and is underway. And it's been exacerbated because we're in a time of crisis. And so there has been considerable scientific uncertainty. There have been conflicting recommendations, for example, that we heard very early on when we started doing this work. You know, the CDC, if you all remember, first recommended do not wearing a mask. And then that was changed later on during the course of the pandemic. And from a scientific perspective, you know, that means that science is working. We're able to test different approaches. We're able to see the results of those different approaches. And then we're able to update recommendations to really reflect that evidence. The other piece that we've really been struggling with is that I think many individuals and many communities that we have spoken with have continued to try to adhere to all of the non-pharmaceutical interventions that have been recommended. So wearing a mask, social distancing, staying home. Unfortunately, we're still seeing in this country, as you all know, huge rates not only of prevalence of the virus itself, but we're also seeing huge individuals of people dying. And so that has been very concerning for individuals. The other piece that we have very much struggled with regards to vaccine acceptance is the issue that politicians and not scientific experts have really been the public face of crisis management. We've heard from a lot of communities that we have spoken with, even just the idea and the name Operation Warp Speed does not necessarily convey or connotate sort of a trustful and safe process. So with regards to some of the things that we have been doing in terms of looking at how do we look at vaccine acceptance, so how do we make sure that as the vaccine is rolling out, we're able to convince people to get the vaccine. What I'm showing you here is essentially the share of US adults who said that they would get the vaccine. And you're looking at this from all the way to the left, starting at the end of February, to all the way to the right at the end of September. And what you're essentially seeing here is if you're seeing the dark green line, that is all adults, there is around 65% of adults said in early March that they would be willing to get a vaccine. Unfortunately, what you can see is in September, that had dropped quite considerably, at least about 10 percentage points. And this change and this discrepancy with regards to willingness to get a vaccine is even more marked if you look at women versus men, for example. So we actually have been seeing a decrease in confidence. And so the question really becomes, what is driving hesitancy? Why are people concerned? Why are people waiting to get the vaccine? So if I can provide a little bit of context here, is that pre-COVID, in terms of why are people not getting vaccinated, there were several arguments that we heard from individuals that really concerned them and led them to either delay or reject vaccines. So those pre-COVID arguments were individuals were concerned about vaccine ingredients. We heard a lot of individuals, particularly parents, were concerned about the vaccine schedule, meaning they were concerned about the number and the different types of vaccines that their children were getting. People were concerned about vaccine side effects. And there were also low levels of risk perception, meaning people did not think that they were susceptible to the pathogen that the vaccine is designed to protect. And they also did not feel as though that the pathogen was severe enough to warrant a vaccine. Now, this has changed a bit with regards to what's happening with COVID. We are seeing huge amounts of distrust, and this is distrust towards government. This is distrust towards public health in general. This is distrust towards healthcare providers. We're also facing an infodemic. There is a ton of misinformation that is being circulated, particularly on social media. And we're also struggling with a polarization of attitudes. So I'm going to talk briefly about each of these things. So in some of the work that I have been focusing on is we've been really looking at specific groups and trying to understand how hesitancy has changed with regards to specific groups. The chart that I'm showing you here essentially shows you the percentage of US adults who, again, told us, essentially said that they would get or not get a vaccine if it was free and if it was deemed safe and recommended. You can see that there's huge disparities here, and this is comparing September to December, but there's huge disparities when you break this down by race, with individuals that identified as Black or Hispanic less likely than individuals that identify as white being willing to get the vaccine. You can see that has also changed over time. With regards to misinformation and disinformation, so dictionary.com in 2018, their word of the year was misinformation. And what we're really seeing is that when people spread misinformation, they often believe the information that they are sharing versus disinformation that is crafted and disseminated with the intent to mislead others. The best example I can give you here that has happened within the COVID-19 context is that, let's say a political leader claims that COVID-19 is no worse than the flu, despite knowing otherwise, then that is disinformation. However, when an individual hears that information, believes it, and then shares it, that is misinformation. And that is one of the main reasons that we're seeing huge levels of hesitancy related to the vaccine. The third, I think, main driver that has been really, I think, highlighted within the COVID-19 context is the polarization of attitudes. So what I'm showing you here, and this was from Nature, is essentially a snapshot of a cluster of individuals on Facebook. Essentially, the dots that are red are those that represent people that are anti-vaccine views. Those that are in blue are pro-vaccine, and those that are in green are typically those that are undecided. And what we have found is that attitudes are very much clustering. Compared to 10 years ago, in terms of looking at vaccine attitudes, there was much more mixing between the blue dots and the red dots. Unfortunately, we're seeing much more polarization, which is making it harder and harder to talk to, let's say, a neighbor or a family or a friend that might not be so pro-vaccine if you are pro-vaccine. So how should we communicate about the vaccines? What are sort of the things that we have learned and that we have been testing? So as I mentioned, trust is an underlying issue that we have been facing. And so it's really important through all levels of individuals that are included in the response, whether that's individuals such as myself that are in public health, those that are working for healthcare institutions, political leaders, community leaders, all of these different types. It's very important to communicate to build, maintain, and restore trust between the public and those that are managing the outbreak. And as Dr. Moss mentioned, this includes being very clear and transparent about communication regarding side effects so that people are able to make an informed decision. The second piece I would say here is announcing early. So really using something called proactive communication, even with incomplete information. So we've been working closely with the Baltimore City Health Department with regards to their response. And one thing that we very much focused on is when the Baltimore mayor gave his press briefings and continues to give his press briefings, he's very clear on this is what we know, but this is what we don't know. Transparency goes hand in hand with both of these two issues. Again, disseminating trial data, talking about that trial data, also discussing and being very clear and articulating how decisions are made with regards to allocation can go a long way. And finally, I think the piece that we have been really trying to focus on with regards to increasing specific pockets of acceptance is really being empathetic. We're living in a very uncertain time. Individuals feel as though their lives have been turned upside down. This has been a prolonged pandemic. And so understanding public risk, perceptions, and views is really critical, and being empathetic is very, very important. A couple of other key points that I will make here that we've learned much more in the clinical setting, I would say, is that if you're trying to talk to an individual, a loved one, or a friend that does not want to get the vaccine, it's very important to not correct misperceptions. We have tested this, and we have found that if you do correct misperceptions, this tends to lead to individuals essentially really digging their heels in, if you will, and not really being open to hearing your argument. The other piece that's very important for acceptance here is focusing on the disease. You know, it's very unfortunate, but here in the United States, now most people essentially know someone that has been either affected or infected with COVID. And most people now, sadly, again, know someone that has likely died from the disease. So focusing on the fact that people are susceptible, that it's a severe disease, is very important. The other thing that we have found is really using something called presumptive communication. So really talking to people about how vaccination is the norm. And for the most part, that is exactly true. Most individuals accept vaccines. Most people believe in the science of vaccines and are clear with regards to the benefits that vaccines can confer. And the last thing I would say is, again, to my last point on my last slide, is really building trust through empathy. I think this goes a long way when we're talking to community members, when you're talking to friends and family that maybe have different views than you do with regards to vaccines, is really being empathetic and understanding that people are essentially trying to do what's best to not only protect themselves, but to protect their families. Thank you so much for giving me the opportunity to talk to you all today.
Thank you, Dr. LeMay, for addressing our doubts and fears about COVID-19 vaccines. Your presentation increased our trust in getting the COVID-19 vaccines. Our next speakers are Dr. Denise Benkel and Pauline Ferranti from the New York City Department of Health and Mental Hygiene. Dr. Benkel works for the Department of Health and Mental Hygiene's Bureau of Immunization. She is involved in contact tracing and currently works in the vaccine section, securing allocation of COVID-19 vaccine to special populations. Ms. Ferranti is the Manhattan liaison for the department's Office of External Affairs. Both of them will now speak about how many vaccines New York will get and how they will be distributed to the public.
Okay, so I'm supposed to share my screen now, right? Okay. Can you see my screen?
Yes, Dr. Benkel.
Everybody can see COVID? Okay, great. Okay. Okay, everyone can see that. All right, hi, I'm Denise Benkel from the New York City Health Department, like I was just introduced, and I'm very happy to be here sharing some information about vaccine allocation and distribution. First, a little bit of a background. Currently, we are very concerned about cases and the % positivity. If you look at this graph on the right, you see date is on the x-axis, October 11th, October 18th. These represent weeks, and the y-axis represents number of citywide cases, and you see the steady increase. So very concerning, and we don't see any plateau of it really yet as well. On the left here, this is a map of all the different areas in New York City, for the boroughs, and then here is Staten Island. and the percent positive, the darker it is, the greater the percent positive is. And you can see some sections are particularly of particular concern. Now, since March of this year, when we had our first our first experience with the disease, we were having 7-day averages and total cases per day that was elevating until about April, May, and then they started to go down. And we had it was down for a number of months, but as you see, we started to go up in November, and there's really no end in sight in total cases per day, or as the dark line shows, the 7-day average. So obviously of great concern. We need to help stop the spread. We are advising people to get tested and separate from others immediately if they test positive. You can go to nyc.gov/COVID test or call 212-COVID-19 and you can get information about getting tested. Also, to stop the spread, you can follow the core 4 prevention strategies. First, stay home if sick, or if you tested positive for COVID-19, if you recently traveled out of state, or if you've been in close contact with someone who has had COVID-19, has it currently. Second prevention strategy, wear a face covering when outside your home, whether you're indoors or outdoors. And actually, I think once you get used to doing this, it's almost like when you go out without the protection, it feels weird. So once you start wearing that mask every time you go out, you literally feel naked when you're outside without it. The third core prevention strategy is to keep 6 feet of distance from people who are not members of your household, who you're not in a pod with. And finally, washing your hands or use hand sanitizer as much as possible. Another thing to stop the spread is to avoid travel and gatherings. This was repeatedly explained by Dr. Fauci around Thanksgiving, around Christmas. It was heeded by some people, not by other people. And we did end up with increases in cases within an incubation period after those holiday events. Finally, follow Commissioner's advisory for older New Yorkers, people with underlying health conditions, and their household members and caregivers. Now, the goal for the vaccination campaign is vaccine for all. There's a New York City Vaccine Command Center, which includes the New York City Health Department operations. I'm going to go into detail on the next slide. The vaccine campaign New York City works with state and federal partners. It focuses on equitable distribution, including acknowledging the history and current experiences of racism and bias in medicine. Finally, the campaign partners with community, elected, city agency, and sector partners. So the Vaccine Command Center, it's a New York interagency effort. It involves the New York City Health Department, Health and Hospitals Corporation, Emergency Management, Racial Inclusion and Equity Task Force, and it all coordinates vaccine distribution in New York City. It procures and distributes vaccine throughout New York City in accordance with federal and state guidance. It executes comprehensive community engagement and outreach with an emphasis on equity. It monitors key data points, tracks progress, and identifies gaps in operations. Finally, it educates New Yorkers with science-based information and prevents the spread of misinformation, like was just discussed in the last presentation. Since there is not enough vaccine available all at once, vaccine is being distributed in phases. The CDC Advisory Committee on Immunization Practices, or ACIP, sets guidance for the country, and New York City and New York State's plan is based on these recommendations. This approach to phased distribution of a vaccine is rooted in equity and will prioritize individuals who have been hardest hit by the pandemic. Who will be offered vaccine first? Well, people at increased risk of getting COVID-19 or at increased risk of severe COVID-19 illness are being prioritized for early vaccination. People who work in high-risk healthcare settings, such as the ICU or emergency room, and residents and staff of long-term care facilities were eligible to receive the vaccine first, along with first responders in medical roles. Recently, additional populations have been made eligible for vaccination, including most healthcare workers, medical examiners, coroners, and at-risk funeral workers, as well as frontline public health workers with patient contact. As you see here with all the listing of those eligible for vaccine, the one of most interest to a lot of people right now is people age 65 and older. It was 75 and older for a little while, but then it went to 65 and older. We expect essential workers who can't physically distance and people at high risk of complications from COVID-19 because of underlying medical conditions, that they will be prioritized next, though the exact details are still being worked out. For up-to-date eligibility groups, visit nyc.gov/COVID vaccine distribution. There's a lot of information on our site. If you go to nyc.gov/coronavirus, you can get to any site from there, or if you specifically go to the site that I just read out, you can get the most up-to-date eligibility group. When will vaccine be available to everyone? Well, New York State determines how groups are prioritized for a vaccine and the timeline for distribution. We actually have to follow what New York State gives guidance, tells us to do. And a lot of things within the health department, New York City is always a separate entity from New York State, but we usually can have our own guidance. In the case of this COVID-19 vaccine distribution, we're really beholden to what the state determines for next prioritization. The phased distribution will take time, and we don't expect vaccines to be widely available until the middle of this year. The New York City Health Department is overseeing vaccine distribution for the city to ensure broad and equitable availability. New York City allocates additional vaccine on a weekly basis. The exact numbers are decided by the federal government based on availability, number of people needing to be vaccinated, and other factors. Through a centralized database, we're able to see which healthcare facilities need more or less vaccine, and we place orders for a vaccine based on need and storage capacity of the individual sites. Where will I be able to get vaccinated? The city has started opening vaccine sites for eligible people and by appointment only. Some of these sites are urgent cares, hospital-based clinics, some pharmacies, specific pharmacies, community health centers, also known as federally qualified health centers, some medical offices, and there are also New York City hubs, which we know in the health department as PODs, point of distribution, which are these mass vaccination clinics, usually held at schools, usually in their gyms. And also the New York State and the Health and Hospital Corporation are holding what we call these mega pods at places like the Javits Center and I think even Yankee Stadium will be holding these mass clinics. And some of them will actually be going 24 hours. Now, if you see nyc.gov/COVID vaccine distribution, you'll get information on who can be vaccinated and where, and for information on how to make an appointment. This is the same site I mentioned on the last slide. So check the New York City Health Department's webpage regularly for updates. How can you help right now with what's going on with the pandemic? Well, keep getting tested and practicing the core 4 prevention strategies that I mentioned on that earlier slide. Help New Yorkers access accurate information by newyorkcity.gov/COVID vaccine and newyorkcity.gov/VCC. Again, if you can't remember anything else from my talk, nyc.gov/coronavirus can get you to all the other information on our website. Share information on social media. Use our social media kit, http://on.nyc.gov/COVID-vax-social to amplify these important messages, and use hashtag New York City Vaccine for All and hashtag I got the shot NYC. Help amplify the word. If you know someone who has been vaccinated, help them share their story and spread the word. Thank you very much.
Thank you, Dr. Benkel, for bringing us up to speed on when we can get the COVID-19 vaccine.
Thank you, Dr. Benkel. Just one correction about the baseball stadium. It's actually Shea City Field.
Oh, City Field, sorry.
I just dated myself and called it Shea Stadium, so now you know how old I am.
Oh, I never can remember the name.
As a lifelong Mets fan, we always call it Shea.
Yes, that is true, Jesus. I agree with you 100%. So thank you so much for having me tonight. I'm following Dr. Benkel from the Health Department just to share with you a little bit about the city's plan for rollout. Currently, there are 794,650 vaccine doses delivered to New York City. As of this morning, 303,671 have already been administered, both dose 1 and dose 2 combined. That big numbers for those 2 doses. So I want to touch base a little bit about the concerns that we have been hearing at the Health Department with regards to accessibility of getting appointments. I know that that's one of the biggest concerns that folks have been alerting us over the past few weeks since rollout has begun. And I wanted to let everyone know again that the city is working very hard to make appointments available. Again, it's based on the availability of the vaccine and the accessibility about scheduling appointments online. We understood that not everyone has access to the internet, which is why we opened up the vaccine hotline at 877-VAX-4-NYC. And that is scheduled to be a 24/7 hotline pretty soon based on feedback from the community that the mayor heard. So the mayor did announce that that would be 24/7 soon. But again, with regards to appointment availability, we really cannot stress enough that it's based on how many vaccines we have available in the city. And for those who are eligible to receive them, that they're properly distributed and allocated to those folks. Also, with regards to locations of our vaccine hubs and other places where people can go and receive their vaccine, we understand that there has been a lot of concern for seniors, particularly homebound seniors who cannot travel very far from their home. And we would love to be able to stand up sites in every single location that is suggested to the city. We welcome suggestions, but again, the one caveat about this particular vaccine, both the Pfizer and Moderna vaccine, is the storage component. So it does require refrigeration, and because of this storage capacity concern, not every location that is suggested to the city as a pop-up site or a mobile site is possible. And so we want to be able to manage everybody's expectations. We do understand that there are many NORCs, which are naturally occurring retirement communities in Manhattan, large buildings that now contain many, many older adults. My parents live in one in Chinatown, Confucius Plaza. And they are very, very interested in bringing the vaccine directly to the seniors who live in these buildings. And while we are very interested in holding these types of vaccination clinics, again, the capacity issue to allow for social distancing, and also that additional 15 to 30 minute wait after you receive your vaccination is a concern as well. So a venue needs to be very large to promote social distancing, safe distancing among all those who are waiting to get their vaccine, and then also a space where people can sit and wait 15 to 30 minutes after they receive their vaccine, just in case there are any adverse reactions. In addition to that, like I mentioned, the storage situation for the vaccine itself. And so because of those issues, it's been a little bit difficult for us to identify more locations within communities to bring the vaccine directly to folks that need it. However, it's not impossible, and we're working very, very hard to make these happen. So as the vaccine becomes more available, as the city has more ability to obtain refrigeration that's appropriate, these are all possibilities in which we can start opening up more community-based vaccination sites. Additionally, we're working very closely with pharmacies, especially large box scale pharmacies that are very common in all neighborhoods throughout Manhattan. And so hopefully they will be online very soon. And so people will be able to access vaccines through pharmacies. And again, we encourage everyone, if you have a trusted physician, please call your doctor's office and see when they will be getting their allocated doses so that you can work with your doctor directly and you don't have to go to a vaccine hub to get your vaccine. You can work with your primary care physician or trusted physician in your family in a location that is comfortable and safe for you. So all of these things I wanted to address to let the city and Community Board 6 especially know that we have heard all of your concerns. We are working through a lot of these capacity issues and storage issues. And we are doing our very best in making sure that this vaccine is accessible to everybody across the city. And in an effort to not take up too much time, I will allow for the next speaker and we'll be happy to answer questions. Thank you.
Thank you, Ms. Ferranti, for clarifying and giving us information on the distribution of COVID-19 vaccine. Our last speaker tonight is Dr. Sabrina Feldson. Dr. Feldson is at the NYU Langone Vaccine Center. She will now speak about the clinical trials that have gone on for the COVID-19 vaccine, as well as safety and efficacy regarding those vaccines.
Hi, thank you so much for having me. Just checking, everyone can hear me and see my screen? Great. So I am a primary care physician at the VA hospital in Manhattan is my clinical location, and also a teacher of medical students and residents at NYU. And with the COVID pandemic, I was invited to be a co-director of our of the VA subsite of a vaccine center, to found that center under the guidance and leadership of Mark Mulligan at NYU Langone Vaccine Center. And some of my slides are repetitive and I'm gonna zoom through those thanks to the presenters before us. But one thing leads right into this, what I wanted to focus on mostly, which is the question of how can you help right now? And I wanted to make a plug right there for participate in a vaccine trial or other COVID trial for monoclonal antibodies or other trials that are available here in your backyard at NYU, at Bellevue, at the Manhattan VA, all within Community Board 6. So we would love to have you participate. The NYU Langone Vaccine Center has worked on the Pfizer vaccine. We are currently probably closing tomorrow for the AstraZeneca vaccine, COVID-19 vaccine trial. Our next slotted vaccine trial, probably beginning in February, is a phase 2/3 Sanofi trial. And so at the end of this, there'll be an email and a QR code where you're welcome to screenshot that and give us a call. So thank you for inviting me. The NYU Vaccine Center is part of 9 or 10 VTEUs. These are NIH-funded vaccine and treatment evaluation units. They've been around since the early 60s, moved around to different academic locations as the places that are sort of easily have access to large populations and expertise to conduct these big clinical trials. And our NYU Langone Center is working as one of these VTEUs. They're the recipients also of the Operation Warp Speed, some of those contracts as a site for those. We're also part of the CoPVN network, COVID-19 prevention network, also an NIH group with expertise in infectious disease from around the country. The hub is in Fred Hutch Cancer Center in Seattle. And these folks and we're part of a weekly calls and networking and ethical issues and questions and concerns around these big trials as well. So we're meeting weekly with them as well. The way that Mark Mulligan at NYU has decided to operate as a VTEU is to have the NYU Langone as a hub for his center, and then open these satellite sites that you see at the bottom here. So the VA is one of them, HHC Bellevue, and there's a Winthrop and also Brooklyn Hospital that's part of NYU that is one of the subsites in order to have the trials open to multiple communities and a diverse population. Just as with vaccine hesitancy, we see a lot of trial hesitancy. It's real. Many of the issues are the same as with getting a vaccine, but vaccine development depends on all our trial participants. And I bet there's some of you out there, potentially even on this phone call. So thank you if you have participated already in one of our clinical trials. Here is a picture here on with the masked gentleman is Dr. Michael Saylor, who's an infectious disease doctor at the VA for many, many years. The chief of medicine actually now retired, but he was in the AstraZeneca trial at our site. One cause of hesitancy is that we see is a concern about can one of the vaccines, can they give you COVID? And they cannot. And this is just a slide. Most of you probably know this, but pieces of a bicycle do not make the bicycle, just as the pieces of the virus here that are used in the vaccine do not equal the virus. They're not infectious. Many of these slides, as you see, are from CoPVN. The vaccines work by teaching your body to recognize and fight invaders. So instead of, if you get SARS is a foreign invader. Many people concerned about using a vaccine as putting something foreign in your body. SARS itself is foreign. Our body sees foreign things all the time. And we'd like to get the foreign thing to be the vaccine rather than the actual virus, because the vaccine and the virus sound a body alarm, fighter cells and proteins go into action. And ideally with the vaccine, you can neutralize it, as Dr. Moss talked about earlier. So let me quickly go through these. The vaccine candidates differ in the message they send, all related though to the spike protein and how that message is delivered. And that's called the different vaccine platforms. In the AstraZeneca trial, as Dr. Moss said, was a non-replicating chimpanzee adenovirus, the common cold virus with the DNA inside. And then Sanofi that's coming next is a protein virus. A protein platform, more traditional platform. All of the vaccines go through 4 main phases of during a clinical trial. Some of these have been put together during this pandemic, lumped together in order to expedite things. But they started all with a phase 1 where we had 20 to 100 healthy volunteers asking, is this vaccine safe? Are there any serious side effects? And how does the vaccine dose relate to any side effects? So you can figure out the optimal dose to use. Then you move on to phase 2 and phase 3. Phase 2 with several hundred volunteers, and here are some of the main questions. And our vaccines now that we've been trialing are mostly in phase 3, and here it says 1,000 or more volunteers. And the COVID vaccine, one of the ways that they've expedited the development is to have not 1,000 participants, but really 30 to 45,000. I think Pfizer at 45 or so thousand participants. So very, very big studies. And that's helped to speed the process without this compromise. So we're doing phase 3 trials, and there's now, we're entering phase 4 as well with some of the trials, some of the vaccines that are have these emergency use authorizations, the EUAs, where we're continuing to monitor longer term side effects that we could not, with the vaccine trials where they had gotten at this point. I think there was, it was a maximum of 6 months, 4 months, I think, after the second vaccine. So we just don't have long-term data yet, and we are continuing the trials and continuing to follow for any adverse events. Volunteers in a trial, it's a very regulated, very procedural, very monitored situation. So clinical trials testing the safety and effectiveness of a new vaccine, you first will go through an eligibility screen, looking at your baseline health and whether or not you meet eligibility criteria and in order to participate. You'll be followed very, very closely. At your first visit, you can expect an exam and blood work and a nasal swab. You'll follow, in most of these, they're 2-year trials. So that's what the EUAs, the emergency use authorizations that we have, are those, the Pfizer and Moderna are still in trial. So the phase 3 trial is still ongoing. They hit, their EUA approval is based on something called an interim analysis, which was a preset time or preset data point when they hit enough COVID-19 cases within the trial to do an analysis of the data and get some efficacy data. But they are still ongoing. They're 2 years. And even though most side effects from vaccines happen within, I think, the first month or certainly 3 months, they're still being followed for 2 years. We need to know durability data. Do we need a booster? That's all really important, and that's all going to require more time. So during the study, you're very well plugged in with the study staff and physicians. And after the study, follow-up safety monitoring. So participants are compensated for every on-site and many telephone visits as well. So there is a compensation for time and travel also for participation. And you're continued to be monitored, but much less frequently over the last year of the study. It's super important to keep our healthy volunteers safe, and vaccines in general need to be very, very safe. We're giving them to healthy people and pregnant women and children. The bar of safety is very, very high. And we saw, and so you're monitored very closely for adverse events during a trial, and you must report any adverse events, whether they're more of the reactogenicity, the type that we expect from your immune system reacting to the vaccine, as Dr. Moss pointed out, those very common effects from the vaccine and expected really effects, to unexpected negative or harmful things that occur. And this happened, we saw when a serious event happens, we saw what happens in the trial, because the AstraZeneca trial had a case of a neurologic issue in one of their volunteers in England. And that caused a pause in the trial. and the trial was paused, I think, for 6 weeks here in this country as the as data was reviewed on all adenoviruses and in an unblinded way. A data safety and monitoring board, an independent board, reviewed all of the data before it could then be resumed. And then what happens is that during the study pause is that all the participants in the study are notified. No vaccine is given during that pause as the data is being reviewed. And then a determination is made. Might this be from the vaccine product? May it not be? And if it is, potentially, even though we don't know, it's 1 out of thousands and thousands, a case that could happen even without the vaccine. But if it potentially is from the vaccine, it then gets built into the informed consent as part of the informed consent for our participants. How do we test a vaccine to see if it works? The participants are randomly assigned to one of 2 groups. This can be in a 2-to-1 ratio for every 3 people to get the vaccine and 1 the placebo. AstraZeneca is like that. So you'd have a 67% chance more or less of getting the vaccine versus 33% of getting the saline, or in a 1-to-1 ratio where you have a 50% chance of getting the vaccine versus placebo. Now, the emergency use authorizations are going to impact this. While randomization will still occur, it's going to be difficult to randomize people ethically to placebo when we have vaccines that have an EUA. And so we suspect that in the future studies, Sanofi even, they're grappling with what are we going to randomize to? How are we going to do that? Because, and they're thinking more in terms of a non-inferiority study, looking at the study vaccine and comparing it instead of to placebo, maybe to one of the EUA vaccines. What does it mean for a vaccine to be effective? It means that this isn't happening. What we saw on the Dr. Benkel's slide here of COVID going up, up, and up. When vaccines have been introduced in the past, when they're effective, they make just a very dramatic impact on prevalence and incidence of disease. So we want that. In our studies, participants complete follow-up visits, usually about 6 to 8 over the 2 years. They get tested for blood. In the blood, they're getting tested for antibodies to the spike, the neutralizing antibodies to the spike protein, and also antibodies to nucleocapsid and one of the other 29 proteins in the COVID-19, in the SARS-CoV-2 virus, because that can tell us, is somebody getting infected but not having symptoms, and that's a clue of whether or not you can be infectious as well. The participants who develop symptoms during the trial come in immediately for an illness visit. We then confirm whether or not they do have COVID, and then they are monitored closely, ideally at home during that period. We collect then, this is the way that the efficacies, this is what the efficacy that's been reported, the 95% efficacy that we see for Moderna and Pfizer so far, the approximate 70% with AstraZeneca and in the combined trials in England and Brazil. This is how it's calculated here with the equation on the bottom. But basically, it's talking about efficacy of preventing symptomatic COVID, because the way that these folks in yellow appear here is if they call and say they feel sick, and then we swab them and then we have COVID. So we're looking at the number of people who get COVID in the active vaccine group and comparing it to the number of people who get COVID in the placebo group. And that gives us the efficacy. So 2 out of 50 here versus 8 out of 50 here. And then with our equation, we get a 75% efficacy for this particular vaccine. I'm gonna skip this, but basically just a reminder that the EUA of approval is different than how normally the FDA approves a vaccine at the end of its phase 3 trials, in the sense that it is based on this preliminary data of the pre-specified numbers of symptomatic COVID And when that occurred, in the case of Moderna, it was 196 cases. In the case of Pfizer, it was 170. Then they analyzed that data in the way that I presented before, and they tell us there's a 94 to 95% efficacy of vaccine and decreasing symptomatic COVID. And also, very importantly, there weren't cases of serious COVID in the people who were vaccinated. The serious cases occurred in people who were not. I think almost exclusively. Certainly in Moderna, also I think in Pfizer and in AstraZeneca. So really, that's really amazing. So we know that this is just reinforcing that we have very good data and are very, very grateful for the vaccines for reducing symptomatic COVID and also for reducing severe disease. We're still plugging vaccine trials. We think it's really crucial. We need more vaccines of more types. This is also from CoPVN. So we need more vaccines that are easier to use. As several people brought up, the freezing issue, as Pauline Ferrante was mentioning, that even if we have the doses, they're not usable everywhere. Because of the freezing. And this is even more of an issue if you're talking internationally, globally, how to get these negative 80, vaccines that require negative 80 temperatures distributed globally. But even rurally, locally here, even within the city, it's an issue. Also, all of both of the EUA vaccines require 2 vaccines. The Johnson & Johnson is 1 vaccine. That's a huge, really eases the burden of vaccinating large numbers with 1 vaccine instead of requiring 2. So more vaccines we have available, and with these different sort of challenges that they each have, the better. Also, we need vaccines that can work for many kinds of people worldwide. So racial, people of color, older adults, age 65 and older. We have some good evidence of that in some, in Pfizer and Moderna. I can't remember which, there was a trial that, I think it might have been J&J that's, or no, I think it was Sanofi actually, that has went back to the drawing board in terms of dosing because they didn't see the efficacy in the older population that they wanted. So they were playing a little bit more with dosing. And also with people with pre-existing medical conditions. And I want to add to this, I think that, yes, we need vaccines that work for people worldwide, but we also need vaccine trial participants who look like all these people to be role models for our population in the city, our population nationally, our population worldwide, to say, hey, I'm in there, and so I feel better taking this vaccine. If you don't see yourself represented, it's less compelling. Also, we still have questions, and all of the speakers have alluded to this, but we have really important questions that need to be answered, including, can the vaccine prevent infection with coronavirus? So you can still have it in your respiratory cells, in your nasal cells. You may not be symptomatic, but you may be infected with it, and then you can still spread it. Also, will we need a booster? How long does the vaccine help the immune system? How long are our B and T cells, our antibodies active? And if we do need a booster, when do we need a booster? This is going to take time to sort out, and we just don't know that yet. Even with our EUA, Pfizer and Moderna, we don't know that yet, but we do know a lot. So here are some of the questions, and I think that Dr. Moss also pointed some of these out. I'm going to move on. Because we're still in this state of, we have something great, but it's not perfect. We need to wear our masks. We need to wash our hands. We need to maintain social distance. Vaccines are not a replacement for those things, until everybody's vaccinated. And so we need to continue to protect each other with these very basic and important techniques that we have here. And we can see this out of our, right off of FDR, the old smallpox hospital from the 1850s that is now in ruins, but so sort of fascinating over there on Roosevelt Island. But thanks to the vaccine, the smallpox vaccine, this hospital is now decaying and crumbling. And as healthcare providers and researchers and community members and global citizens, we hope to partner with you to defeat the epidemic and the pandemic. And we hope to hear from any of you who are interested in participating in any of the trials. I wanted to thank you again to CB6 and to the NYU Vaccine Center. A big thank you to Mark Mulligan and Manor Ali and Hannah, who all helped me so much with these slides. And here you see the QR code. There's a registry if you're interested in hearing more. These are the 5 subsites listed on the right. If any of you are veterans, we would love to have you at the Manhattan VA at the bottom there. Just call that number. Thank you so much.
Thank you for your presentation, Dr. Felson. So we will now turn to committee members who raised their hand through Zoom, as well as questions from the audience and that had been submitted through the QA feature of Zoom. Presenters, committee members will unmute themselves to ask their own questions. Once committee members are done asking questions, Kyle will read the questions aloud from the Q&A feature of Zoom. So please feel free to answer questions as appropriate based on your area of expertise.
All right, so let's begin with Marty. So Marty's hand up. Marty, you're still on mute.
There we go. Throughout the development of the vaccines, the marketing kept being, oh, this is the fastest it has ever been. You know, the mumps took 4 years or whatever it was. Since SARS in 2002, the teams have been working on these vaccines. Why has the media been shown that, oh, it's done in 10 months? It hasn't. It's been years that the various teams have been working on these. Granted, COVID-19 is a little bit different than bird flu or SARS, but nonetheless, it's the same processes that they've been working on since 2002.
Yeah, I think I can say a little bit about that, which is if you do listen to TWIV, This Week in Virology, or, you know, I can't remember her last name, Kumikia. People have been, you were right, they have been working, Moderna vaccine, the mRNA vaccines have been worked on for at least a decade. And I think it's because the phase 1, phase 2, phase 3, phase 4 of the vaccine trials have been condensed. And that's kind of, you know, that in between sort of the epidemics, people would start to work on other things. You know, SARS didn't blow up as you know, it was contained and burned out. MERS the same. And so they would get some momentum and then it would sort of die out and they would work on other things. And I just think the pace of the phases and the combining of phases was you know, wasn't sort of dissected in the media in a way that was helpful for inspiring confidence. I don't know, Dr. Moss has been at it and may have more to add to that.
Yes, I think the timeline that is often referred to in the media is the time from the identification of a novel virus, and SARS coronavirus is different than other coronaviruses. Agree, there's a similarity, to the time that we have an emergency use authorization, and as Dr. Felsen said, completed phase 1, phase 2, and phase 3 trials. That this is a remarkable timeline for that, and so I'm not sure I would agree that we've been working on a vaccine for this virus for a long time. There's been work on messenger RNA platforms, yes, for a long time, and for adenovirus vector platforms for a long time, and that's what made it possible to develop a vaccine for SARS coronavirus 2 in such a short time, because the platforms were there, and those genetic platforms are very flexible platforms. So a new pathogen emerges, it's very easy to kind of put in the genetic recipe and adapt it. But what made as Dr. Felsen said, there are other things that made this process faster than usual. The combination of phase 1 and phase 2 components of the trials, the combination of phase 2 and phase 3. But I'll say the most important thing in my mind, or one of the most important things, is that in a pandemic, one can see very quickly the difference in the efficacy between the vaccinated group and the placebo group, because there are so many cases. The cases are accumulating so quickly that you see that difference quickly. And outside a pandemic, that just takes a long time. So the fact that we were in such a terrible pandemic in the United States facilitated our ability to draw inferences about a protective efficacy very quickly.
Thank you.
All right, thank you, Dr. Moss. Are there any other questions from the committee or any of our panelists? All right, so just a reminder to attendees from the public, please use the Q&A function. We will not be calling on people who raise their hands, so please do submit your questions through the Q&A. As we let our attendees put their questions in, we do have many questions for our speakers tonight, so we will be succinct and to the point because we do want to respect our speakers' time. The first set of questions will go to Dr. Felsen. There are a lot of questions about the work that you're doing. So since you just spoke, I'll address those now. So the first is, what are the NYU priorities for getting the vaccine into the arms of those in phase 1B, particularly those who are over the age of 75?
So the NYU is relying a lot on, and for NYU patients is relying a lot on MyChart. And for the folks who are eligible for the vaccine, and they're using over age 65 as well now, they are sending a personalized MyChart text to your phone. And I think I had been talking to Hannah, who's on the Hannah Wade, who's on the phone now or online now, who thought there may be a phone number, maybe there will be one coming, because for people who aren't using MyChart for NYU for the vaccine. But I think the best way also is getting in touch with your local primary care provider, or through the city as well, through the what Dr. Benkel put up as well.
Thank you. So furthermore, in particular, are the volunteers who are receiving the placebo then given the vaccine at any point? Are the volunteers told what they have received in a blinded study, and when would they be told?
Yeah, so this is a big area of conversation within this co-PVN weekly phone call, and I can tell you what we're doing now. So, and Moderna and Pfizer have already had to grapple with that, and AstraZeneca, and now that they are available, that's why I'm saying the EUAs are going to really impact studies going forward, and what's going to be done with that placebo arm. And different studies are doing different things. By the way, Hannah Wade is going to follow up with the NYU specific information on that first question. For the trials, what I think, and again, Dr. Moss may know this more specifically, but I think both Pfizer and Moderna, for their participants who were in the placebo arm, they crossed over and they received, were offered the vaccine. I think that they were, I don't know if they did, I think one of them did it in a double blind, and it kept them blinded. So everyone who got the placebo got the vaccine, and everyone who got the vaccine got the placebo. I think another one opened it up and just said, hey, you got the placebo, here you get the EUA, and they're following still for the 2 years for safety. It changes the endpoints, it changes what's being looked at, but they're continuing in the study. AstraZeneca, which we are enrolling in until tomorrow, but we've gotten many requests for unblinding from the participants because they are many of them are in groups where they are offered Moderna or Pfizer and they want to get them. They want the vaccine. And what AstraZeneca, what we're doing is we are unblinding people. We can do it within 24 hours usually, unblinding them and letting people know who are eligible for the vaccine or have an appointment for Moderna or Pfizer. And if they got placebo, we are recommending that they get Moderna and Pfizer or Pfizer. And if they got the AstraZeneca vaccine, even just the first dose, we're recommending that they stay in the trial and get the second one as well, because we really don't have any data on what mixing the vaccines do. And then we're recommending if you go out and get one of the EUA vaccines to continue in the trial also for safety. So that's how they're dealing with it in AstraZeneca. And I think in the studies that are still to come, I don't think there'll be placebo.
Okay, thank you. And then as a follow-up to that, which dosage of AstraZeneca is being given as a first dose? The mistake half dose that led to a higher efficacy rate, or the lower dosage that you intend to give?
So AstraZeneca's had some issues, and in the half dose was given in Britain as the accident, and what's being done in the 30,000 people who are in the trial in the US is the full dose followed by the full dose. It's not the half dose. That they felt like they wanted, that had already been in process here. They wanted to keep that data clean and see what happens. Maybe there'll be a study afterwards, but it would be a new design study. So they're doing the dose here is the full, okay, not the half.
And then finally, when it is your turn for a vaccine at NYU Langone, which Langone centers are available for patients to receive a vaccine?
I don't have the specifics on that, so I will have to get Hannaway to get back to the group and post it. Can you post it afterwards somewhere?
Sure, yeah, we can follow up. For anyone whose questions are not answered tonight,
Yeah.
please follow up with our board office. The email is info@cbsix.org.
I think that's evolving, is my understanding, and maybe Pauline can also respond to that. NYU, because I think that specific NYU doctors also in the community will get them. I think all of the major hospitals, NYU affiliates, have it. Certainly NYU Langone.
Okay, thank you so much. Dr. Moss, we have some questions for you as well. So one of our attendees said that in your presentation, you said you could still get the virus even though you've been vaccinated. Is the virus less virulent if you have been vaccinated? And if so, how much less?
Yeah, so it's not, the vaccine doesn't change the virulence of the virus. What happens is the body's immune system, the disease is different in our body. So I just want to kind of clarify that. It's not a kind of a change in the virulence of the virus. It's a change in what the virus does to our body. And I think Dr. Felsen kind of alluded to some of this too. So we, there were no cases of severe disease in the vaccinated group. So we think that those people who do get disease, COVID-19 disease following vaccination, tend to have milder disease, we think, and we'll learn more about that as the trials are followed. What is unknown yet from the phase 3 trials, although we'll also learn more about this, is whether vaccinated individuals can get asymptomatic infection. But I think the short answer to this question is that yes, if an individual gets infected after a vaccination, they're more likely to have milder disease than severe disease.
Thank you. And as a follow-up to this, actually, we have several questions which are along the same lines about, I guess their phrasing is percentage of protection. So after you've taken the vaccination, first of all, what would be the percentage of protection? Is there a way to determine what your personal percentage of immunity is? And is there any additional immunity gained by taking another series of these vaccines several months later?
Yeah, so these are somewhat complex questions. There's no way to assign at an individual level kind of a percent protection. We do that at the population level. So a vaccine efficacy of 95% means 95% of a population we would expect to be protected. That doesn't apply to the individual level. I think going forward, as we learn more about the protective immunity, it may be that a certain antibody level following vaccination is is correlated with protection, and so it may be possible to say more about how well a person's protected based on their antibody level. Whether that's going to be, that'll certainly be done in studies, and Dr. Felson's studies are certainly looking at antibody levels and what we call correlates of protection. Whether that's going to be available to the general public, I don't know. We don't traditionally measure antibody responses after we vaccinate people, in the general population. So I would say yes, that 95% doesn't provide doesn't, sorry, I lost my words, lost my thought, doesn't apply to the individual. But we think that antibody levels, particularly what are called neutralizing antibody levels, will be a correlate of protection, and that is theoretically at least a way an individual could assess their own immunity.
Right, thank you. And then again, more questions for you, Dr. Moss. The vaccines that we're talking about, are they protective against the new COVID-19 strains that we're hearing about? And if a booster shot is needed, based on your experience, what would the estimate be for that? Would we need 2 shots or 1? Or if you could just repeat what you had discussed about boosters.
I'll start with the second one. I mean, the bottom line, and Dr. Pelson talked about this, because we're still so early, really, in the phase 3 trials. As she said, these trials follow individuals out for 2 years, and I'm sure there'll be, other studies that follow out longer. We really don't know the duration of protection. So we really don't know anything about, when a booster dose would be needed and how frequently. We can only just really guess in part based on the duration of immunity following other coronaviruses, but whether that applies here or not is not clear. So the real honest answer to the duration of immunity and booster doses is that we do not yet know yet. We're still very early. And so now I'm going to have you remind me of the first part of the question. Sorry.
The first question was about, sorry.
Yeah.
About if the vaccines that we're discussing right now, does that account the new strains that we're hearing about?
Yes. So, this is a complex question, really. And so the bottom line is that all viruses mutate. And so it's not unexpected that this virus has mutated, particularly given how much it's transmitted, which provides more opportunities for mutation. There are ongoing studies right now to see whether some of the particular mutations in this virus, and going back to what we talked about, there are mutations in the spike protein that we're really concerned about, because that's what the vaccines are targeting. So are there particular mutations in the spike protein that I would say decrease the efficacy of the vaccine? And so again, the honest answer is we do not know yet. There are studies, and what I've read is that we'll have results from some of those studies very soon, within the next week or two. That'll give us some indication of whether these variants are, whether our current vaccines protect against these variants, or at least they're more neutralizing studies. They're more kind of laboratory studies. I do not expect that we will see marked decrease in the protective efficacy of our current vaccines against these variants, but we'll just have to see. These vaccines actually induce antibody responses to multiple parts of the spike protein, and so a single mutation, the spike protein would have to change a lot. And there are certain constraints on how much that spike protein changes. And just put it back into the terms that we talked about. If the spike protein changes so much, it won't be able to bind the receptor on our cells and get into our cells. So that's a dead end for the virus. So there are constraints on how much that spike protein can change. And that also limits its ability to evolve away from our immune responses.
All right, thank you, Dr. Moss. Dr. LeMay, we have a question for you as well. So particularly when talking to a vaccine doubter, is it helpful to bring to their attention of the problems of long haulers who have contracted the virus?
It's a great question. I think for me, I would really focus on what is the severity and susceptibility. So we don't want to scare people would be my only hesitation in doing that. I think being very upfront about severity and susceptibility of the disease is good, as long as we're not scaring people.
Fantastic, thank you. And then I have a question for the group. We've received several questions about this in terms of vaccine distribution, sort of the policy behind it. Are there any conversations, or could you provide more insight into how the vaccines will be distributed and administered to people in developing countries, countries with less economic power, less infrastructure, maybe even less data as to how many people are actually infected by the virus?
I can start with that one, Kyle.
Okay.
This is a really important question, and we think about equity in the United States in terms of how we distribute the vaccine, which is critically important. But we also do need to have a conversation and discussion about global equity around the vaccine. We do know that that a small number of wealthy countries have purchased through bilateral arrangements with the vaccine manufacturers a disproportionate number of vaccine doses, more than half of them, which does limit the ability of other countries around the world, particularly low and middle income countries, to access the vaccine. Fortunately, there is a global initiative called COVAX that's led by the World Health Organization and other partners to ensure that there is some global equity in vaccine distribution. And they've set up a financing mechanism and made purchase agreements with a number of different vaccine manufacturers to ensure that there will be a sufficient number of doses, not for all populations globally. That's going to take some time. But what they're trying to assure is at least a sufficient number of doses for 20% of the population for all countries that basically join this COVAX, which is almost all countries except the United States and Russia, I think. And so there is a global initiative, but I do think the United States has a responsibility to the global community. This is a global pandemic, and I think our commitment will change shortly for the better.
Thank you, Dr. Moss. This next question I'll again open up to everyone. So the question is, should I get tested before actually getting the vaccine? Should I see if I have the virus before actually getting the vaccine, and to determine if I'm symptomatic or asymptomatic?
I mean, I can start with that there, and Dr. Felsen can answer too, maybe. But there's no need to get tested before you get the vaccine. And I assume you mean the questioner means tested for the virus.
Mm-hmm.
One could also ask, get tested for antibodies before the vaccine. But you know, the current recommendations are that even people who've had prior COVID-19 get vaccinated because of the uncertainty of the immune response following natural infection. So there's no need to be tested for the virus or for antibodies prior to vaccination.
All right, thank you, Dr. Moss. Dr. Felsen, do you have
I
anything
was
to add
just
to
gonna,
that?
I heard somewhere in the ether that talk about potentially people who've had symptomatic COVID getting just one vaccine, and maybe that ultimately would be enough in combination with the natural infection and providing long-term immunity. We don't have any idea. That may be something that's studied along the way, and would then free up more vaccine for other people. And I think it's an interesting question, but I don't think we know. And one other thing with the other question about global equity, it's another plug for the vaccine trials, because that's for instance, like AstraZeneca or a one dose vaccine instead of two are much more accessible globally, and also much cheaper. For whatever reason, I think AstraZeneca is $4 per per dose versus 20 or something for Moderna. I may be messing that up a little bit, but there's definitely a cost differential also in the vaccines.
No, you're exactly right. The AstraZeneca has deliberately set a low price. They're not making profit off the vaccine in an attempt to get the vaccine to low and middle income countries. They also, I think, Dr. Felsen, as you know, the AstraZeneca vaccine has much less stringent cold chain requirements. It's really just refrigeration rather than freezer. So it's very it's very compatible, I'll say, with current vaccine distribution refrigeration requirements.
All right, thank you both. So Pauline, I'll turn it to you. I see that you wanted to answer one of the questions from the public, particularly about adverse reactions. So please take the floor.
Well, I'm not a medical doctor, so I can't speak particularly or specifically about adverse reactions, but I did see a question about what would happen in a vaccination hub should an adverse reaction occur. And so built into the city's structure, we do have medical professionals on hand to be able to monitor everybody who has received the vaccine, which is why we ask everyone to stay for the 15 to 30 minutes after they receive the shot, should they need medical attention. Of course, then they would be asked to go to the hospital as well. So I don't know if any of the MDs wanted to clarify sort of what specifically that would look like.
This is largely driven by, and Dr. Felsen will know, but largely driven by the anaphylactic reactions, the severe allergic reactions that were first reported in the United Kingdom when they rolled out the Pfizer vaccine, and then we've seen cases in the United States. That reaction was not observed during the phase 3 trials, at least during the preliminary. During the phase 3 trials. So that was kind of an unexpected reaction, but the people are advised to stay. First of all, the FDA has required that these vaccines be administered in settings where they can handle an anaphylactic reaction and have the medications to do so, such as epinephrine. And that waiting for that 15 to 30 minutes is to allow time to see if there is an early severe allergic reaction.
Perfect, thank you. And actually, as a follow-up to that question, Dr. Felsen, our audience wanted to know if there were any adverse reactions identified in your trial, and what were they? Was it as described by Dr. Moss?
Well, we're not doing one trial. As I mentioned, the NYU, the vaccine center was involved with Pfizer. Our first trial right now is the AstraZeneca that we're closing, and then we'll be doing Sanofi, but they've done others, monoclonal antibody studies and others also at the vaccine center hub. We've seen some, mostly the reactions that we're seeing are the reactogenicity type things, probably a little bit less I'm saying this totally off the cuff, the data, I don't know yet, but then with Moderna and Pfizer, in terms of that, you know, how some people describe feeling flu-ish type symptoms, either the sore arm or just feeling like a cranky baby for a day afterwards, like maybe some muscle aches and even low-grade fevers. That's been the main thing, gone within, you know, 24 hours or so. That's by far been what we've seen, by far and away the most we've seen. There was with AstraZeneca in England, there was a severe adverse event that was identified, a neurologic event, a transverse myelitis, an ascending weakness that paused the study for six weeks here while data was reviewed. There was also a person who died in Brazil in the AstraZeneca trial, but that person was in the placebo. As the trials get big, 30,000 people, adverse events happen. Adverse events are unexpected harmful events that may or may not be severe, and they may or may not be related to the vaccine, but they're all recorded and they're all followed. And when one of them is considered serious, there's a pause to look at, really to identify whether or not it's related to the vaccine, and then to document that and understand that as best as possible, as deeply as possible, I guess. But things happen to 30,000 people in a trial unrelated to the vaccine as well. And those are also looked at and evaluated. So there was this neurologic event, and that it's still unclear. It hasn't been, it hasn't been seen again. Yeah, that's what I can say about it now. I don't know if anyone else.
So actually, based on that, another question that someone asked was about a particular situation if this were to come up. Say you have your first vaccination and then are compelled to receive your next dose at another venue where the original vaccine that you took was not available. Can you match that up with a new, the second dose of another manufacturer, or does it have to be from
That's what we're really the professional advice of co-PVN and the infectious disease doctors and folks talking around this is not to do that, not to combine one vaccine manufacturer vaccine with a different one if it's a 2-dose requirement, because we just have zero data on it. We don't know what the efficacy is going to be. We don't know about adverse events in that context. And so it really, the recommendation is to stick with what you got for the second vaccine. I mean, if you're in a different state, still stick with that one if you can. So the recommendation is really to stick with one. And that, the AstraZeneca is now doing their interim analysis right now. I think it was 150 cases of COVID amongst the participants that was required. It happened. They're doing their interim analysis. They may well also get an EUA soon. And the recommendation is to stick with what you got. That they're all good.
Okay, thank you. Yeah, you heard it here. Please don't mix and match your vaccines.
Yeah.
Now for the city, for Department of Health and Mental Hygiene, we have a question about how many vaccines New York City actually gets, and on a weekly basis, if that's how it's done, and how many people are in the categories are actually eligible.
That's a great question, and I don't have the specifics for that, so I will have to, unless Dr. Benkel, do you have?
I don't know that either.
Okay, so we'll have to get back to the community board on that.
Okay, perfect, thank you. So some other questions that we have for Department of Health is about the actual process. So aside from the actual capacity of vaccines, why is it that it's so complex and tedious? We've heard that some people have spent, many hours trying to get an appointment. Every location has its own specific registration form, which you fill out to find out that there are no appointments. Is there some way to make this process simpler and easier for users?
I wish we had a simple answer for that, but it really is specific to each location and the allocation of doses that they have. And so there it really isn't a way to integrate all of these different systems into one, because we just don't have that technological capacity to do that. And also each location has a very specific amount of dosage. And so when they're making appointments, it's very specific to their location. And so we understand the frustration, and we are asking folks to please continue to call, continue to check online, because as locations are getting more shipment in, those appointments will start to open up again. And so we really are hoping that people don't get absolutely frustrated with the process. We understand that it is frustrating, and we wish that we had that ability to integrate everything into one, but at this time, we just don't have that capability.
And we have some more for you, Pauline. So in your presentation, you mentioned that a lot of this process is dependent on conversations between the state and the city. Can you provide any insight in terms of who will next be eligible for these vaccines? So we've received some specific parameters, particularly family members of medical workers, if elderly spouses of vets who have been vaccinated already, and yeah, family members, again, of frontline patient-facing medical workers.
Sure. So all eligibility requirements are determined by the state, and we try to update our website as quickly as possible. So on Monday, the governor had announced that it was open up to 70 people, age 75 and up. And then within 24 hours, it was changed to 65 and up. And so we want to make sure that we're giving all New Yorkers accurate information. And so I encourage everyone to continue to check our website, but the actual determination of eligibility is determined by the state.
All right, thank you, Pauline. Another question is about the actual storage of the Pfizer vaccine. So given that, as you mentioned, a low temperature storage is required for that particular vaccine, how can the public feel ensured that the vaccine has been kept at the appropriate temperature and is safe for use? And that is a city question as well, but it's also for the rest of the panel as well about, and perhaps Dr. LeMay, you can address this, but particularly how public confidence can be assured when there's such stringent requirements for how it actually is held.
I believe Dr. LeMay had to hop off, and I would defer to the medical professionals for this particular question.
I mean, one thing I understand is that it comes in batches in dry ice, and that actually that's keeping it cold, and you have to use that within, I think it's within 6 hours. It may be shorter, but there's stringent rules about when you open it, how long you have to use it, and it comes packaged in a certain way. Once the seal is broken, you can't send it to any other facility. That's part of why you were saying you can't do it in an apartment building necessarily. It's not allowed to move. It's tagged or in some way, so you can't move it. But I don't know if anyone else has more details about the.
There are temperature monitors associated with the vaccines. It's actually been one of the big advances in vaccine kind of distribution globally are these kind of temperature monitors. So I think the public can have high confidence that the vaccines are kept at the appropriate temperature. And there are warning, the temperature is very carefully monitored, and there are warning signals if that's not the case.
And it can't be shipped to anywhere if you don't have the capacity to keep it, to freeze it at that time.
Add dry ice. I mean, you can add dry ice to the way it comes for a certain amount of time if you can use the vaccine up, but.
So that was more directly what the question was about, more about city resources. Does the city have the ability to ensure that it will remain effective and properly secured? And if there is additional resources needed, that can be readily available to the city?
But the city doesn't really get the vaccine. We allocate the vaccine. We make a determination each week on who's going to get the vaccine, and we direct it there. So, I mean, we virtually get it, but we really don't get it. I mean, so we're relying on the federal government is really dispersing it, and then it's going directly to those sites. And like it was just said before, you can't even get the vaccine if you don't have the right storage. You have to have shown that you have the right storage capacity for either the ultra cold or the freezer with the Moderna vaccine. So, but I mean, I don't know if I would say that like there's 100% chance, there's like no chance that anything ever happened to it. I mean, you have a certain amount of trust, I guess, in any kind of cold chain storage you have to have when you get any kind of medical care from a provider. You know, it's not like everybody's taking this very seriously. I think everybody who's vaccinating is taking it seriously, and they're not leaving it out, or if it is out, they're throwing it out. So I think we have to have a certain amount of faith in the system too, and it deservedly so. I think that, you know, people understand the gravity of this.
Thank you. And actually, our last question of the night is actually addressed to Dr. Benkel, but also open to the rest of the panel. Is it guaranteed that when we schedule the second dose at the time we're given the first dose, that it will be within the time frame of CDC recommendations?
There's when you schedule it, well, it would be, yes, it would be within CDC recommendations, but you have to understand if you get it delayed, it doesn't affect the efficacy, how good it is. It just delays it. So you don't really, I don't want people to worry like, oh my gosh, if I don't get it by that day, it's not going to be good. That's not true. It'll be just as efficacious. It'll just be a little bit delayed. But there's a process set up so that second dose can be given on time though. But not to worry if it's a little late.
All right, thank you, Dr. Benkel. And do any of our panelists have any final words before we conclude tonight? Just want to give a final opportunity to make any comments you'd like to make. I don't see any other questions from the public.
Dr. Moss and Dr. Levine already signed off.
Okay, thank you. All right, so let's conclude. So I want to thank again our wonderful speakers tonight. It really means a lot to Manhattan Community Board 6 and our communities and all of the public attendees who attended for you to have taken time out of your very busy schedules to inform our communities about the COVID-19 vaccine. And then before we end tonight, I would also like to thank our Health and Human Services Committee Chair, Dr. Elvie Barroso, for putting this program together, as well as to our board staff, District Manager Jesus Perez and Community Associate Brendan Berth for their coordination and organization. And I also want to thank, I saw them online earlier, I want to thank in particular Manhattan Community Board 8, our family to the north, the chair, Alita Camp, who just recently transitioned out of that role, as well as District Manager Will Brightbill, who was a terrific partner in getting this information out to the public. So thank you very much. And then lastly, I want to thank all of the attendees for your participation, thoughtfulness, and your care for this subject area. I hope that you will now take this information going forward and share it with your networks. This program has been streamed on YouTube, so it's there for ease of shareability. So please do send that out to your networks. And then again, I would like to encourage everyone once again to continue to attend our Health and Human Services Committee meetings, which take place at the second Thursday of every month. So the next meeting will take place at 6:30 p.m. on Thursday, February 11th, 2021. I hope that you can all attend. So good night, everybody, and I hope everyone stays well. And thank you again for your participation. Have a great night.
Thank you.
Yeah.